Hello guys, let's begin with the new lecture today. Today we'll talk about photobiology and how exactly the plant respond to the different light condition. Let me tell you that the plant has both senses, like they can sense the quality and the quantity of the light. Largely the physiological mechanism, even the developmental biology of the plant is dependent on the light, its intensity, its quality, its quantity, how long a light is going to be there. So everything is going to affect the overall process in the plant, both the photomorphogenesis as well as the physiological mechanism in the plants. It's a warm welcome from Spice Up Your Biology, Sandeep here. Let's talk about the pigments which senses the light's quality. So that is today's topic, and name of the topic is phytochromes. So phytochromes, these are the pigments that controls the growth and flowering in many plants. Even the many other physiological mechanism that is governed by the phytochrome will not talk about the many other things, especially the seed germination or the many other physiological process. So today we'll be focusing more on the part which is related with the flowering of the plant. So what are the activities that is governed by the photoregulation? Before I go into the detail of the structure and all, so there'll be a few things which we keep in the mind. Photosynthesis, it is largely dependent on the light quantity. Photomorphogenesis is dependent on the light quality. Photomorphogenesis means the development of the plant, or the how exactly the body part of the plant is going to be formed in the presence of the light. For example, if you do not give proper light to a leaf or germinating seedlings, the color of the leaf may not be the green, or it is going to be attenuated. Third thing is the photoperiodism. This is largely dependent on the light duration. It will be a focal point today. And phototropism, how the plant they respond in particular direction to the light. Photoregulation. So another part is that the pigments which are going to be involved in photosynthesis, we have the three pigments: photoreceptors, chlorophylls, and carotenoids. And other part is going to be photosynthetic efficiency, which is the part of the regulation that is going to be regulated by the light intensity. Is going to be high or is going to be low, and it has been governed by the immersion enhancement effect, or the red drop and other thing that will not be talking about today. What are the photoreceptors overall the plants are going to have? So plant has a receptor which can sense the light in the various way. So there are the few classes of the photoreceptors which we are going to talk about. Let me remind you that all the photoreceptor has a two part. One is going to be the pigment which can respond to the particular color of light, and the second thing is going to be the protein which directly or indirectly, or it can be a signaling molecule which can give the signal in order to bring the metabolic changes in the plant physiology. So first is going to be phytochrome that we'll discuss today. Is a red and far red, the two substitute or the two alternatives will be existing in the plant. Second one is a cryptochrome, which largely absorb the light in the blue and the UV irradiation. They do play important role in the settling development and the flowering as well. Third one is going to be phototropin, which will play important role in the differential growth of the light in the particular gradient way, which will be responsible for absorbing the blue and the UV irradiation. Fourth one is the UVB receptors. We do have this, but still we do not have many information about it. Let me begin with the phytochromes. As I told you, like they respond to the two different kind of light, a red light and far red light, and there'll be two alternative form. But let me remind you here, more studies that has been done on the phytochrome, both the monocots and the dicots. It has revealed that the phytochrome might absorb the blue range as well. blue light can be absorbed by the phytochromes. There'll be 5 genes in the dicot because of 5A to 5A, 5A, B, C, D, E, whereas the 3 A, B, C in the case of monocot or a rice plant, which has been discussed, which has been studied in the due course of time. Let me first just begin. What is a phytochrome? As I told you in the beginning, it is a pigment found in the plant cell, basically seated in the leaf, where it controls the development of the plant and the physiology of the plant. More importantly, the flowering of the plant. We'll go just little in the mechanism as well, with the within few minutes. The pigment, as I told you, has a 2 phases. It can exist in one form which is sensitive to the red light, another is the far red light, and they are interconvertible. So actual plant response is very specific to the each of the species that is put under the studies, and some plants which are uh, completely neutral. So we cannot just generalize the fact that every plant is dependent on the light, especially in order the some physiological activities. So there can be certain plant, they may not be affected, their flowering may not be affected due to the light. Let's see the bigger picture. So both the form of the pigment, PR or PFR, they have their own structure, which is very much similar, and they're interconvertible. If you just look at, I was just talking about, you can see these part, which we can see the several rings, we call it as a phytochrome or the phytochromobilins, and they're connected with the protein part, which is shown in the light green. So here they can exist in the two forms, cis and the trans form, and the conversion of one into the another will lead to the several physiological activities. Just look at how this phytochromobilin had come from. So heme, which is unlikely to be present, in the plant, that will broken into the biliverdin, which is again found in the serum protein or in the biles. And this is broken into the phytochromobilin. The structure is such. So this is the one, and these entire activities happening into the plastid. And again, they will connected with the protein, and that protein is having other activities which is to be done. They have different domains that is said to be the histidine kinase, or it could be the serine, threonine kinases. There's a lot of discussion over it, but for the time being, we'll just take it. So we have the two form, you can see over here, the cis and the trans, and they convert one form into the other, and this is the phi domain of it. So which are the wavelength are photoperiodic? Photoperiodic, a term of photoperiodism, it was discovered by the Allard and the Gardner, and they were uh, talking about the length of the light, which is very, very important. So just here, when we talk about the length of the light, we have to be focused on either of the part, the light part as well as the dark part. If you see into it, so phytochrome pigment tends to revert to the PR during the long periods of the darkness, and under the light condition, they'll be converted into the PFR. Thus, the length of the exposure of the light in a building or it's outdoors, which play a very important role, in the conversion of the one form of the light into the other. Then we have the photoreversibility. There's one basic experiment has been carried out where it has been shown that the effect of this, they have taken the lettuce seed where they have given the 3 hours, prior to the irradiation time were red 1 minute, far red 3 minutes, and germination was recorded. Look at over here. If just giving the red light at the 22 degrees centigrade in darkness, so what is happening? The germination rate is going to be 88%. Now, if you give it the far red, immediately after the red light, the percentage of the germination will decrease to 22%. Again, if you give the red light, it will increase to 84%. If you followed by the far red, it will just jump down to the 18. Again followed by the red light, it is at 72%, and followed by the red light, far red, it will again go to the 22%. This suggests, and they have a trend. The trend is that the red light is conducible to seed germination. Now you see another, the structure that I was talking about a little more biochemically. If you look at these points, one is that, if you see the PR and you see the structure, this is the cis isomer. Look at the D ring, and if this reminds you of the ring of the phycocyanine and the phycoerythrin, where we see the tetrapyrrole ring in an open structure. In the chlorophyll, this structure is going to be closed, and in the case of phycocyanine or the phycoerythrin or in the phytochrome is going to be open. Look at this. This is going to be, if you see, for just your understanding, this is on the lower side, so it's going to be cis, and if it goes up, you can see over here, so this is going to be trans. The basic connotation is that, in this cis, the groups are, you can see these two groups are coming very close to each other, whereas in trans they are little far from each other. So red light converts cis form of the PR or cis form of the phytochrome into the trans form. So cis form is also called as PR, and trans form is also called as PFR. And you can see on the left hand side, these are the chain of the amino acid. This is the protein part. One more basic mechanism which is related with the signaling. Sometime we'll have a complete lecture over the signaling part of it. Today we'll just confine to the basic information. Look at over here. If the nights are short, so what happens is that this FT, or you can see from over here, this is the leaf on the either side. This is one which is the Oryza sativa, a rice plant, which is a long day plant. Long night plant or the short day plant. Another one is a short night or the long day plant. Look at what is happening. Leaves, they get the light, the clock genes or the early phase genes, they will make the constant CO, a transcription factor. Later on is going to be converted to the protein. FT messenger RNA will be formed, and from there the FT is going to be created, and this FT will go to the suprapical meristem, the SAM region, and from there they get the various information whether the flowering is going to happen or it is not going to happen. There are certain changes which is going to happen. Similarly, if you look at over here in the case of Oryza, and all the movement is through phloem. Look at the another part where we have the long night. In that case, what is happening is that the SD3A is going to be formed that will travel along to the SAM, and then that affects the flowering in the Oryza plant. Let's talk about the further more information about the photoperiodic responses. It is all about the preferences. Long day plant flower when there is a prolonged larger pool of PFR, and the short day plant flower when there is a prolonged larger pool of the PR. It is very clear to you, like if the light is more, it's going to be PFR. If the light is lesser, it's going to be PFR, which is going to exist there. Look at over here. This is the range 660 nanometer, and the red light is given, PR is getting converted to the PFR. So this is the synthesis which is going to happen, and this is the destruction. So here is going to be reproductive flowering, and here are going to be vegetative phases. If you look at over here, mid-summer sunlight at 660 nanometer, PR is going to be created, then the PFR is going to be created out of it, because of the red light, and again the flowering is going to happen. So long plant need or sense low PR to bloom. So low PR required for the long day plant, and the high, the PFR or the let's the see in another part, the long day plant, since the PR. Look at what happened is that if the PR is there, it's going to be vegetative phase, and if it is a PFR, is going to be there more, so it is going to be reproductive. So PFR, if it is more than PR, it is going to be the flowering which is happening in the long day plant. Another thing you can see over here is that in the if you see the sunset or the far red light, which is around 740 nanometer, this is creating the PFR to be converted to the PR. So again, what will happen is that the flowering is going to be affected. Now let's come to the salt day plant. This is midsummer sunlight, which is at 660 nanometer. PR is going to be converted into the PFR. So PR is the one which is here responsible for the reproductive flowering, whereas the PFR is one which is required for the vegetative phase of the plant. Plant usually exists in the two forms, the vegetative or juvenile phase and the reproductive or the adult phase, where the flowers are going to be formed. So salt day plant eventually require the low PFR. Low PFR means high PR. Got the point? The low PFR means high PR alternatively. So in winter, far red light is going to be more. So there'll be more conversion of the PFR into PR. So flowering is going to be affected. Salt day plant need again the low PFR. Similarly, the night, the PR and PFR level is going to vary. This is what I told you. So this is how the day and night cycle is going to be very, very important. So synthesis and the reversion help the tell the time. This is how the plant comes to know. If I tell you the fact, some plant they flower more in the summer, some plant they flower more in the winter, or some they flower in the rainy season. I'm talking about the Indian conditions. So this is how this phytochrome is going to play a very important role. Which one is going to be more decides whether the flowering is going to take place in that particular condition or not. So elegance of the photoperiod, plants not only can differentiate the wavelength, we look at over here 660 and 740, and alternatively they can convert into the PR and the PFR form, and the ratio of the forms that will change over a period of time depending upon the light length or the dark length alternative. So the more the PR, there'll be more flowering in the salt day plant. This is very, very important phenomenon that you need to know. And you can interlink this with, if you require PR more, that means the darker period is going to be more. So that is where you can easily decide which weather it is going to flower. So salt day or the long night, maximum PFR conversion is going to happen. Just now I told you, this is how the PFR and the PR interconversion works. Then long day plant need the sense low PR. Low PR will be only when the period of the light is going to be longer. So they'll be having more of the PR getting converted to the PFR. And if the darker condition at 740 nanometer, if it is going to be coming, then the flowering chances are going to be more. So low PR means more PFR. Low PR means the long day. High PFR means the short day or more longer period of darkness. So this is again the ratio. Long day means short nights. Short nights means less PFR and more PR. So short night means the PFR level is going to be more compared to the PR, because long day convert PR into the PFR. Look at this. This is going to give you some idea. Long day, short night, no flower. Short day, long night, it is going to flower. If we are having the short day and the interrupted the darkness with the light, here you can see the presence of the darkness sparsity is going to be very, very important here for the salt day plant. If you just entrap with the white light, there'll be no flowering. Even if you interrupt with the red light, there'll be no flowering. But if you are interrupting with the red light and just followed by the far red light, means the flowering is going to happen. Probably you'll be able to connect what I was talking about. So salt day plant has a single cycle. That is, the example is going to be rice. Long day plant, single cycles, example is a dill. Day neutral plant, they will not be affected. Cucumber, amphio, ambiphototropic plants like Ceracia, these plants are inhibited by the intermediate day length. Short then long day plants like the clover. Similarly, the qualitative long day plants like the hibiscus, most of the constant long day to initiate the photoperiodic responses. Quantitative long day plant like the foxgloves. Similarly, the qualitative short day plants in Insidia. So hopefully you have enjoyed this lecture. This has given you some idea how the photoperiodism works. A very, very warm welcome again from the Spice Up Your Biology. Hopefully you enjoyed the lecture. Watch it completely, made notes out of it, and hopefully you're going to share with other and subscribe my channel as well. Thanks a lot for listening so carefully. Thank you.